Well, students aren't joining quickly. Can somebody confirm that you can hear me?
Yeah, we can hear you.
Thank you, Jennifer. All right. Before we begin, are there any questions? All right. Before I share my screen, let me go to my grade book. I've got some things graded. Close that down. I can leave that open. I got Lab 5, Lab 6, and Lab 7 graded for the second time. Lab 9 was graded for the first time. Let me see if you can still work on that. And if you did not like the grade on Lab 9, you can turn it in again until November 30th. Now realize that's Thanksgiving weekend, so I also got the lecture graded. I think I redid lecture 6, so chapter 6 and chapter 8. And I have not done the unknown project for Monday or today, so I'll be looking for that after the lab today. It might be kind of late today, but I hope to get the unknown project stuff out tonight. Any questions about any of that? If not, let me close down the grade book and we will begin. I should probably close down the email too, because if an email comes in, it'll make a noise. All right, any questions before we begin? Let me share my screen if I can find it. Should be seeing my screen now. So today is November 26th. We're going to be discussing uh, chapter 13, viruses, viroids, and prions, getting about halfway through the chapter. There is no lab today. I will be logging in for 15 minutes to answer any questions. You should be working on your unknown project. And then let me remind you, there is no class on Thursday. I think everybody knows that. Happy Thanksgiving. And the unknown notes pages will close Friday, November 29th. And if you request anything on Friday, you have to get it, make it a 1-day test. Because the unknown project has to be turned in by Saturday, November 30th. Any questions about any of that? All right, let's go to chapter 13. I've got that up here someplace. Oh, there it is. I have that we had discussed the general characteristics of viruses. We talked about the host range being the range of cells that a virus can infect. And most viruses only infect some cells of one or a few species, like HIV, can only infect a few human cells as well as a few cells of one of the chimpanzees. It does not affect other species, and it does not infect most human cells like skin cells or gut cells. This specificity is due to the binding of the virus, or it's mostly due to the binding of the virus to the host cell receptors, which are molecules on the host cell that the virus binds to. And we have a term for the molecules on the virus. They're either called ligands or adhesions, depending on who the author is. It's the molecule on the virus that binds to the host cell receptor. Both the host cell receptor and the adhesions on the virus tend to be glycoproteins, but they can be proteins or something else. Um, on the virus, these molecules that bind to the host cell tend to be the outermost molecules on the virus. When we're talking about viruses size, this cell is trying to show you. Let me see if I can view this a little better. Trying to show you the different sizes of viruses. They range from 20 to 440 nanometers, and a average prokaryote cell is about 1 to 2 micrometers or microns, and that would be a 1 to 2,000 nanometers. And so a virus is considerably smaller than most prokaryote cells. And then a prokaryote, as you know, E. coli is actually a little bit larger than most prokaryotes. Um, is much smaller than a eukaryote cell. There would be a red blood cell right there, uh, a little larger than the average prokaryote cell, being E. coli here. And then the virus. The longest virus is Ebola. It's about 1,000 nanometers long, but it's not the largest virus. The largest virus that we're going to talk about here is megavirus, and it's the largest virus by diameter. It's not the largest virus by length, as Ebola is, but Ebola is not very large in its diameter, and so megavirus is the largest virus. Any question, at least the largest one we're going to talk about. Viruses can go down to 20 nanometers. Let me see if I can blow that up and read this. I think the smallest virus is something like MS 3, no, MS2. I'm sure no one's ever heard of MS2, but the smallest virus you've ever heard of is just a little bit larger than MS2. It's the polio virus, 30 nanometers, one of the smallest viruses. When we're talking about virus structure, it's important to realize that we use the term virion as a single mature, complete infectious virus particle. So when a virus replicates, you usually say that it replicates into new virions. Viruses have nucleic acid for their genome, but unlike cells, their genome can be either DNA or RNA. It is never both, it is one or the other, but their genomes can be RNA. And this genome can be double-stranded or it can be single-stranded. Double-stranded DNA is not unusual for our cells. Most of the time, that's what our DNA is. Single-strand DNA isn't unusual either. There are some viruses that have single-strand DNA for their genome. If you recall, our cells are single-stranded. Whenever the DNA is actively being replicated or being transcribed. A double-stranded RNA genome is unheard of in our cells. We do not have double-stranded RNA, but some viruses have a genome that's double-stranded RNA. And then some viruses have a genome that's single-strand RNA, and single-strand RNA occurs in our cells by like messenger RNA or ribosomal RNA, and much of tRNA is single-stranded. tRNA can loop around on itself. Why? It's not always said to be always single-stranded, but it is one single-strand RNA. It just loops on itself. And then when we're talking about a virus that is single-strand RNA, its genome, you need to discuss whether it's a positive single-strand RNA or a negative single-strand RNA genome. A positive single-strand RNA genome can be read by the ribosome, meaning it will have AUG in it, and the ribosome can read it. The negative single-strand RNA is the complement of the positive single-strand RNA. So it will not have AUG in it. It will have the complement. Let me see if I can do that in my head. U, A, C would be the complement of the negative single-strand RNA. So the negative single-strand RNA is just the complement of the positive single-strand RNA. And the positive single-strand RNA is, of course, what messenger RNA is. And the ribosome can read the positive single-strand RNA viral genome. Any question about any of that? The viral genomes can be linear or they can be circular, depending on the species of virus. A linear RNA, or should I say, a linear genome of a virus is normal because our cells have linear DNA for their genome. And then a circular viral genome is normal because prokaryotes have a circular DNA genome. Any question about any of that? And then some viruses, like the influenza virus, have their genome in several segments. This is actually normal for humans because our genome is in several segments. We call each of these segments a chromosome in our genome. We don't usually call the virus genome a chromosome because chromosome includes DNA and DNA and protein, and viral genomes do not usually have protein bound to the genome. And of course, their genome could be RNA as well. Any question about any of that? All right, when we're talking about the viral structure, when we're talking about a virus, it's important to realize that all viruses have a capsid, and that is the protein coat surrounding the nucleic acid. Let me blow this up so you can see it a little better. So this virus has this portion of the virus, the protein capsid cut out so you can see inside the virus, seeing the genome being the central molecule of the virus. And then all viruses have a protein coat surrounding it that we call the capsid. The capsid is made of capsomeres, and you see this little circular thing there? That is a capsomere. Some viruses, like this one, only have one capsomere in the capsid, and it just repeats to make the capsid. That's not always true. Let me show you another slide of a virus. Come on. That, yeah, let's go right here. Here, this virus would have one capsomere making up the head of the capsid, and then at least two capsids making the sheath, capsomeres making the sheath of the virus, and then probably another capsomere in the base plate, maybe another one for the pen, and then at least one capsomere for the tail fibers. So what would that be? 1, 2, 3, 4, 5, at least 6 capsomeres in this virus. Any question about any of that? Some viruses have a envelope surrounding the capsid. Not all viruses have an envelope. An envelope is mostly a lipid bilayer surrounding the protein coat of a virus. Only some viruses have a lipid envelope. Not all viruses do. If the virus has no envelope, we call it a naked virus. If it has an envelope, it is called an enveloped or enveloped virus. Any question about that? The envelope, like I said, is mostly a lipid bilayer, but it does have some proteins and carbohydrates in it. The envelope is external to the viral protein code as well as the viral genome. And so it will be the outermost molecules on some viruses. The envelope may have glycoprotein spikes. Most viruses that have a envelope tend to have glycoprotein spikes on the envelope. Let me blow this up so you can see this a little better. Here's a envelope virus. There's the capsid right there, and there's the genome inside it. Anyways, on this envelope here, we have these spikes protruding out. The spikes tend to be glycoproteins. There are also the outermost molecules of the virus. And so they serve as both antigens. That is something that a animal cell can recognize as being foreign. So like when you get a virus that is infecting you, your host immune system can respond to it. And what your immune system is responding to will be certain molecules on the virus that we call antigens. These antigens are usually the outermost molecules of the virus. The spikes also are the outermost molecules on the virus, and so these are where the adhesions lie on a virus having spikes, meaning this virus, these spikes will serve as the molecules which will attach to the host cell receptor, and they also serve as the antigens that the host immune response will react against. Any question about any of that? When we're talking about a virus, it's important to talk about their general morphology. That is the shape that the virus has. And by shape, we're talking about the shape of the capsid protein outer coat. Some viruses have a helical shape, meaning a helical virus, and there would be a helical virus. This is a helical virus, Ebola virus. I don't see the helix in it, and I'm not sure. Maybe you have to look at the uh, capsid at greater detail. I don't know. And then some viruses have the polyhedral shape, meaning the capsid has a many-sided virus. This is the artist's rendition with this cut away, but a polyhedron, it would be like a fancy dice. And here is a polyhedral virus, actual electron microscopic image. And as you can notice, this virus has a envelope around it as well as protein spikes coming off of it. But the capsid has the polyhedral shape, looking like a fancy dice. The third shape that a virus can have, we call a complex virus, where the capsid is a complex shape. And this shape can be literally anything, like the bacteriophage T4 looks like the Apollo lunar lander. If you remember that from the Apollo spacecraft missions. Here is a complex virus having, I don't know, a bullet type shape or an ovoid type shape. And there's a real odd shape. I don't know what that would be called. This is also an envelope virus, showing you the envelope here, and then the envelope here. Any questions about any of this? So the complex virus can take on almost any shape you can think of. Uh, T4 shown here is a complex virus. It belongs to the T even family of viruses like T2, T4, T6. T4 is a bacteriophage. This is a virus that infects bacteria cells. That's an actual electron microscopic image of one. And there's the artist's rendition of bacteriophage T4. T4 is the most studied virus of all viruses. The reason why T4 was picked to study it in so great detail is that probably because it infects E. coli cells, and E. coli cells were in most microbiology labs. So they just took T4 to study it, and we know more about the virus T4 than any other virus, including human viruses. Any question about any of that? All right, let's talk a little bit about viral infection and the host immune response. When an animal, such as a human, becomes infected with a virus, this will stimulate the host to produce antibodies against the virus. The antibodies recognize and bind to the viral surface proteins, and these we call antigens, what the host immune response responds to. Some viruses can change the viral protein where they change or mutate these virus um, antigens fairly quickly. A good example is actually influenza. Um, with the virus influenza, let me shut this down, show you. I think I have to do it. Nope, I got paint this time the first time. Normally I have to do it 2 times. If you get the flu shot on the first year, so I guess this would be time on the x-axis. After the first year, marking it here, let me change that again. And then this would be year 2, year 3. And on the y-axis will be the immunity. So the percent immunity. When you first get the flu shot, even for young individuals, the immunity will never be 100%. So we'll make 100 like right here. And for a young individual, immunity after getting the flu shot would be probably 95%, certainly 90% or greater. So we're going to start the graph here. And with time, the immunity decreases. After 3 years, your immunity drops to a level where they say that you're no longer protected. Now, this doesn't mean you have no immunity. It just means the immunity is dropped to a level that they say you're no longer protected. How do I word that? Anyways, with enough time, your immunity will eventually drop to 0. I don't know when that would be, but eventually that would happen. But when it drops below a certain threshold, they say that you're no longer protected and that you have a good chance of getting infected with the flu again. My point is that when you get the flu shot, after 3 years, you're no longer being protected from the flu shot. The reason being is because the flu virus can change and its antigens can mutate and change, and after 3 years, the flu shot is no longer good. In fact, in reality, they tell you should get the flu shot every year because your immunity does drop. And so you may not be protected even after the first year. And of course, not everyone is protected even when they first get the flu shot because the immunity is never 100%. It obviously depends on what your age is and what the your immunity is. Individuals who are over 65, for example, don't have an immunity of 95%. I don't know what it is, but it's much less. I believe it's more than 50%, but it does drop. And so their immunity drops to a lower level quicker because you're starting at a lower level. The point is that the flu virus can mutate and change, and then new viruses come out and they will not be protected against the vaccine. Another virus which changes fairly rapidly is COVID-19. Some viruses don't change very rapidly. For example, I'm trying to remember the name of that virus, just a minute. Not the shingles, although that one would be a good one. It's not the best one. Um, I almost had it, starts with a P. Polio. The polio vaccine was made in the 1950s. I think one was made in 56 and the other in 58, and both vaccines are still in use in the world today, meaning polio has not changed since the 1950s. And let's see how long ago that is. That's close to 50 years plus 20 years. So that's like 70 years. And the polio virus has not changed where the vaccine is no longer being protective. And that's actually a good thing. That means when we make the vaccine, it'll last a long time. Any question about any of that? It depends on the virus, how quickly they mutate. Like the influenza viruses mutate very rapidly. And then COVID-19, the coronaviruses, mutate fairly rapidly. I believe the RNA viruses mutate more rapidly than DNA viruses. You don't need to know that. And beyond that, I don't even know. You just have to look at the virus and see how it mutates. All right. When talking about viral taxonomy, people do put viruses into different groups, taxonomies. With viruses, all viruses are grouped into families. A few viruses, in fact, most viruses, I think, are grouped into, is it order? King, Peter, son, Cain, over, from, Germany. Yeah. So, Cain, yeah, order is the grouping above a family. Most viruses do belong to an order, but higher groupings above an order we do not have. All viruses are grouped into orders, or most are grouped into orders, all are grouped into families. They are also grouped into genus. They're grouped into species, and then they are grouped into subspecies. The higher groupings are not made. The reason being is that viruses have a very small genome, and then they evolve very quickly with their host cells that they infect. And so we do not know the long history of viruses. So we cannot group them into something much higher than an order. There is one classification which we do group them in, and that's the not the family, the kingdom that the virus will infect, meaning we do group viruses into animal viruses and plant viruses and fungal viruses and bacterial viruses. So we do group viruses into the kingdom that the viruses infect. We don't know the evolutionary history there, meaning we don't get that from sequencing the viruses. We just look at the cells that the virus infects and then group them into the kingdom. So they are grouped into kingdom, and then they're not really grouped into anything down to an order. They are grouped into families, and a family name always ends with viridae. Like the herpes family is called herpes viridae. They're also grouped into a genus under that, and the herpes Family of viruses would be called the herpes virus or the herpes genus, and the name is virus. So when we're talking about like the herpes simplex virus, you just say herpes um, simplex virus. And actually, that would be a herpes virus for the genus. And then the viral species would be the herpes simplex virus. And that would be a group of viruses sharing the same genetic information and ecological niche, like herpes simplex 1 and herpes simplex 2 both infect human cells. They are uh, similar in their genetic sequence. They obviously infect different cells, or they tend to, although they can infect the same cells. You'll be pleased to know when we classify a virus, we don't have Latin names for them. A virus is named by its common English name. So herpes simplex virus type 1, its species would be herpes simplex virus. Poliovirus, its common English name is poliovirus. There is no Latin name for the virus. And surprisingly, this is used worldwide. French speakers and German speakers, they call the poliovirus, meaning apparently all of the early virologists were English speaking, and they just started naming things and giving them English speaking names. And then the other people in the world, if it's speaking different languages, just used those common English names to refer to the viruses. So the common English names are used worldwide. And then the subspecies of a virus is designated by a number, like herpes simplex type 1, herpes simplex type 2. It's the same species of virus, but a different subspecies. And obviously, you know that the subspecies can infect different cells, like herpes simplex type 1 and herpes simplex type 2. Excuse me here. But the subspecies is even more important than that. For example, poliovirus subspecies 1, meaning poliovirus 1, can give an individual the disease polio. Poliovirus 2, meaning poliovirus subspecies 2, we use as a vaccine to prevent getting the polio disease. So one subspecies gives us the disease polio. The other one is actually the oral live virus, the Sabin vaccine, that we use to prevent getting the disease polio. Any question about any of that? So it does tell you how different subspecies of viruses can be. All right, I've already mentioned that the herpes family of viruses is called herpes viridae. The genus would be herpes simplex virus, the genus name. The, excuse me, that's the species name, herpes simplex virus. I'm trying to remember what the genus name is. Right up, I don't know off the top of my head anymore. Anyways, the subspecies would be uh, herpes simplex type 1 or herpes simplex type 2. Any question about any of that? I will briefly mention, you don't need to know this, that the herpes simplex virus has been given a scientific name, human herpes virus, but most people are not using it, including virologists and the medical profession. And so this change is never taken and it's not sticking. And so I'm just suggesting you learn the name uh, herpes simplex virus, which obviously infects human cells. Any question about any of that? All right, there we're looking at the herpes simplex virus. It can be possible to grow viruses in the lab. For bacteriophages, these are bacterial viruses, you just grow them in bacteria cells. And I even did this where you can plate out bacteria on the plate. Let me get this a little larger. And the reason why the plate looks a little whitish here is because you put bacteria on a lawn on the plate, and so the bacteria will grow everywhere. And before you incubate the plate, you then put on either 1 ml or 1/10 of a ml of a liquid solution containing a virus, and then spread it over the plate. And then you incubate the plate, and wherever there's a virus, it will infect a bacteria cell and then replicate, lyse the cell, be released, and then infect neighboring cells, replicate, lyse the cell, be released, infect more neighboring cell, so that when you see it, you'll see the virus forming plaques on this cell, on the plate, meaning this region, which is white right here, is just bacteria growing in a lawn. And this region here, let's take a real big one right there, that is where there's a plaque, where there was a virus that infected bacteria and then lyse the cells, and the plate is not white there because all the cells are lysed, and it has the normal agar color right there. And that we call a plaque. You can notice different sizes of the plaques, and a large plaque probably is where the virus initially landed on or touched a bacteria cell, so the virus immediately replicated. A small plaque like this one there or that one there probably had the virus landing near a bacteria cell. So here we're going to make a big plate, and here's a bacteria cell, and this little speck here is going to be a virus. And the bacteria cell will replicate, and initially it's still not touching the virus, and then it'll replicate again, still not touching the virus, and then it'll replicate again, still not touching the virus. And this one might replicate, so we'll put another one here. Another one here. And now it replicates again, and here it's finally touching the virus. Of course, it replicates there. My point is the cell had to grow before it started touching the virus, and now the virus can infect the cell. And so this plaque will be smaller than a plaque formed over here, where the virus initially touches the cell and then can infect immediately the cell. Why the plaques can be different sizes, why that plaque is larger than this plaque here. And then, of course, you can also have large plaques when the plaques fuse together, like this looks like it's a fusion of 3 different plaques. Anyways, that's how you grow bacteria, viruses that infect bacteria cells called bacteriophages in a lab, which I actually did. Any question about any of that? When we're talking about viral multiplication, you should remember that the viral genome only contains a few genes, such as the genes encoding the protein coat any of the genes needed by the virus for their replication. And then the virus replicates by hijacking their host cell. Following infection, the virus takes over the host cell's metabolic machinery, and then the virus uses the host cell's ribosomes, the host cell's tRNA, the host cell's amino acids, the host cell's ATP, and some of the host cell enzymes as well. The basic mechanism of viral multiplication is similar in all viruses. For bacteriophages, these are viruses that infect bacteria cells, sometimes called phages or phages. I will always use the term bacteriophages to make it clear that these are viruses infecting a bacteria cell. Bacteriophages have two mechanisms of reproducing: the lytic life cycle and the lysogenic life cycle. A bacteriophage that uses the lytic life cycle to replicate will replicate in their host cell, lyse the host cell, killing it, and then that will release the newly replicated viruses. The lysogenic life cycle, the virus will infect the bacteria host cell, and then the virus and the cell, the host cell it infects, will live together in symbiosis for as long as the virus remains in the lysogenic life cycle. Any question about that? Meaning the host cell remains alive when the virus remains in the lysogenic life cycle. Let me take a step back and say that to make a new bacteriophage, what do we need to replicate? What components does the virus need? Well, the virus is going to replicate. What's it going to need to replicate? Anyone?
Like genetic material, like DNA.
Yeah, it needs to replicate its viral genome. It also needs to replicate its protein code. This is the minimum that a virus needs to replicate if the virus is to replicate itself. So I'm showing you a DNA virus, double-stranded DNA viral genome. It doesn't matter what the viral genome is, it needs to replicate it, making more of the viral genome. It also has to transcribe the viral messenger RNA and then translate that into the viral capsid proteins. And then the capsid proteins need to get together with the viral genome to make the new progeny virions. So that's the minimum we need in viral replication. Obviously, you could have more things required, like if the virus has an envelope, you have to make that and or gain that, I should say, and you may need to make certain viral enzymes. We're not going to talk about that. For the virus following the lytic life cycle, the virus has 5 stages in their life cycle: attachment, penetration, biosynthesis, maturation, and then release. Attachment, the virion has to actually attach to their host cell. So the bacteriophage literally has to attach to their host cell. Oops, didn't mean to do that. That's what I meant to do. Then the virus has to get its genome inside the host cell. This is called penetration. What the virus does is it actually uses the enzyme lysozyme to open a hole in the cell wall, and then the tail sheath contracts, forcing the genome, which is the head in the head of the virus, into the host cell. The virus then will undergo biosynthesis, and this will be the production of the bacteriophage genome, which in this case is double-stranded DNA, as well as the capsid proteins. Those have to be made. And then there's maturation. This is when the newly made viruses assemble, where the viral genome gets together with the protein coat, and the protein coat has to assemble into the coat as well. We'll talk a little bit about the maturation, and that is with viral assembly, like the protein coat, it just naturally comes together. And then the virus has to be released. And what the virus does is, a bacteriophage virus, I should say, will release the enzyme lysozyme, which will degrade the cell wall of their host cell. The host cell will then allow water to come in, and the host cell membrane will expand until the host cell will pop, and that will release the newly replicated virions. That will also kill the host cell. Any question about any of that? If not, let me see if I can get this little movie to go on the viral life cycle.
First, the phage particles stick to the surface of the bacterial cell. They bind to specific sites called
Let me see if I can show you.
In a way, the virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell.
Let me point that out again. The virus contracts. I can't seem to show it.
It is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule.
So the virus actually contracts, and that puts pressure on the head, which actually injects the viral genome into the host cell. Much as a syringe, you put pressure on a syringe, it injects what's in the syringe into the patient. Any questions about any of that?
Is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
All right, any questions about the lytic life cycle? The rest of this movie is the lysogenic life cycle, so I'll hold off on showing you that. We might get to it today. So first you have to have attachment where the virus bacteriophage actually attaches to the host cell. This binding is specific. The adhesions on the virus have to bind to the host cell receptors on the host cell. And that's why one species of virus tends to only infect one or a few cells of one species, although it could be a few species. There are exceptions like equine encephalitis virus can actually infect many different species. I know West Nile virus, which is a member of the same family of viruses, can infect many different species of birds and many different species of mammals. I don't know if it's all, but many, and then can do that. But most viruses only infect a few cells of one or a few species. And then the virus will contract. It actually will digest a little bit of the cell wall right there by using the enzyme lysozyme. And then the sheath will contract. They'll put pressure on the head that'll force the viral genome into the host cell. The host cell, that's penetration. The host cell will then undergo biosynthesis, making the new viral molecules such as the capsid proteins and the viral genome. The host cell, the host cell will be taken over and hijacked to replicate the virus, and then the viral protein coat will self-assemble, meaning once the different proteins are made, they'll start self-assembling into a virus. The genome is then packaged inside the virus, and there's an enzyme the virus has in their genome that actually stuffs the viral genome in the newly made virus. Two terms you should know for the lytic life cycle. That's the burst time. That's the time it takes from infection of the host cell to the time that the host cell bursts to release the newly replicated viruses. For your average virus and your average cell, it'll take 20 to 40 minutes for the virus to infect the cell, replicate, and then burst the cell, releasing the newly replicated virions. This will depend on the temperature, as well as the species of virus and the species of host cell. And then burst size is the other term you should know. This is the number of newly replicated virions synthesized in an infected host cell. Usually this number is between 50 to 200 viral particles. It will depend mostly on the species of the virus, how many viruses will be released in the burst time. Any question about any of that? Remember, the host cell will burst simply by the virus secreting the enzyme lysozyme, which will degrade the host cell wall. That will allow water to enter the host cell until the cytoplasm cell membrane can't expand anymore. It'll pop the cell membrane, and that will release the virions. Any question about any of that? All right, that's enough about the lytic life cycle, where the virus infects the host cell, replicates itself, kills the host cell, which will allow the cell to burst, releasing the newly replicated virions. In the lysogenic life cycle, the virus and the host cell can live in symbiosis for as long as the virus stays in the lysogenic life cycle. when a lysogenic virus first infects their host cell, they have to decide immediately one of two things, and that is which life cycle does the lysogenic virus enter. It can enter the lytic life cycle, meaning a lysogenic life cycle virus or lysogenic virus can enter the lytic life cycle, in which case, if it does, it will just follow the lytic life cycle that I mentioned above, attaching to a new host cell, penetrating the host cell, biosynthesis of the virus, maturation of the virus, and then release of the virus by killing the host cell. And that's the lytic life cycle. It's the same life cycle as the lysogenic. When the lysogenic virus initially chooses to enter the lytic life cycle. But the virus can also enter the lysogenic life cycle, where the phage infects the cell, injects its genome into the host cell, and then chooses to enter the lysogenic life cycle and does not enter the lytic life cycle. When the virus enters the lysogenic life cycle, its genome will be incorporated into the genome of the host cell's chromosome. So this chromosome, let me blow this up. This chromosome contains the viral genome, meaning the reddish purple, and then the purple is the host cell's chromosome. Now, the virus will only replicate whenever the cell replicates, because when the cell replicates its chromosome, it will replicate the viral prophage. And we call the viral genome when it is inside its host cell chromosome, meaning a lysogenic viral genome. We call it a prophage. And it will replicate whenever the chromosome replicates. So the cell will then send the one chromosome to one side of the cell, the other chromosome to the other side of the cell, and then begin dividing down the middle. And this can go on for many cell divisions where the virus and the host cell live together in symbiosis. But occasionally, the virus can leave the lysogenic life cycle and enter the lytic life cycle. For the lysogenic life cycle virus that enters the lytic life cycle, it will first excise the prophage from the host cell's chromosome. And then the separated genome will express the genes for the lytic life cycle. And then the cell will be lysed, the viral genome will encode the production of new virions. The cell will be lysed, releasing the newly replicated virions, which will then infect another host cell. And immediately, once this viral genome is inside the host cell, it have to choose between entering the lysogenic life cycle or entering the lytic life cycle. Any questions about any of that? Now, for reasons that are not understood, every once in a while, a virus which is in the lysogenic life cycle can leave the lysogenic life cycle and enter the lytic life cycle. And this has been studied now for 100 years, and they still don't have a good handle on why the virus leaves the lysogenic life cycle and enters the lytic life cycle. with one exception, and that is if this chromosome gets damaged by, I don't know, radiation or a chemical, meaning if this chromosome gets a mutation, that will tend to switch the virus from the lytic lysogenic life cycle to the lytic life cycle. It's like the virus knows that the host chromosome is damaged and that the host cell may die, and so the virus wants to enter the lytic life cycle to replicate before the host cell dies. Any question about any of that? All right, let's watch I think I've already got it on the video of the lysogenic life cycle.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, everything about this movie
In a way,
is correct,
a virus is a parasite.
with one
It
exception,
attacks a host cell. Each type of phage can only attach to a certain type.
and that is this is showing you bacteriophage T4. Bacteriophage T4 does not enter the lysogenic life cycle. But other than that, everything about this video is correct. Bacteriophage T4 only has the lytic life cycle. Why did I stop the share? Oh, I see why I stopped the share. I wanted to stop this, but we can shut that down. All right. So a virus following the lysogenic life cycle will infect the cell, and then the cell will choose to enter the lysogenic life cycle, where the viral genome will incorporate into the chromosome of the host cell, where we call that a prophage. An important gene of the lysogenic life cycle is the repressor gene, which produces a repressor protein that keeps dormant the genes of the virus to enter the lytic life cycle. If ever this repressor protein is lost or inactivated, the virus will leave the lysogenic life cycle and enter the lytic life cycle. As I stated, it's not clear for most cases why a virus leaves the lysogenic life cycle and enters the lytic life cycle, with one exception, and that is if the host cell has its chromosome damaged, the virus will tend to enter the lytic life cycle because the host cell may die and the virus will want to replicate before the host cell dies. All right, any questions about lysogeny? Let's talk about some of the consequences of lysogeny, meaning if a lysogenic virus enters the lysogenic life cycle with its host cell, that host cell will be immune to infection by the same bacteriophage. So this host cell that has the bacteriophage genome inside it will not become infected with another virus of that same species of virus. So that's the first consequence. The host cell becomes immune to infection by the same species of virus. And the second thing that may happen is conversion of the host cell, called phage conversion, where the host cell can exhibit new properties from expressing the genes in the bacteriophage. The best example of this is when the Corynebacterium diphtherium toxin is made by the bacterium Corynebacterium diphtherium. It can only happen in a lysogenic cell, meaning a host cell, Corynebacterium diphtherium, that has a lysogenic virus infected in it. Because the Corynebacterium diphtherium toxin is encoded by the genome of that virus, the lysogenic virus. Any question about that? So obviously, if you get infected by Corynebacterium diphtherium bacteria, hopefully that bacteria is not infected with the lysogenic virus. Because if it is not, then you cannot get the Corynebacterium diphtherium toxin. But if it is infected with the lysogenic virus, you can get the Corynebacterium diphtherium toxin. Any question about any of that? All right, that's enough about talking about bacteria viruses, also called bacteriophages. We need to talk about the multiplication of animal viruses. Do they have different life cycles or do they have the same life cycles? Oops, went wrong way. An animal virus is a virus which infects and replicates in a host cell belonging to an animal. So animal viruses are viruses which infect the animal kingdom. Animal viruses surprisingly follow the same basic pattern of virus multiplication, but there are several differences. For example, attachment of the virus to the host cell is simpler because it's much easier for a virus to attach to the cell membrane and then enter the cell membrane than it is for a virus to attach to their host cell, which usually has a host cell wall. Penetration is also different, and the animal virus can penetrate the cell by endocytosis or by fusion of the viral envelope to the host cell membrane. Penetration of the lytic life cycle virus, if you remember, was the virus degraded the cell wall, and then the sheath contracted, pushing the viral genome into the host cell. By penetration of an animal virus, you happen to do it by endocytosis or fusion, bringing the virus into the cell. Uncoding of the virus can happen by viral or host enzymes, so that's similar. Biosynthesis will be the same. You have to replicate the viral genome and the viral proteins. Maturation is similar, where the capsid proteins self-assemble, and then the viral nucleic acid has to be stuffed inside the virus. Release can be similar, meaning we do have animal viruses that follow the lytic life cycle, where the virus infects the animal cell, replicates, and then lyses the animal cell to release the newly replicated viruses, meaning the host cell will be killed by the virus, and this we call the lytic life cycle. which is very similar to the lytic life cycle in bacteriophages. But we have actually we have a couple of new life cycles. The main one being the budding life cycle, where the newly replicated virus buds out of their host cell. Any question about that? If the virus follows the budding life cycle, it will have an envelope. The reason being is the envelope is gained by the virus in the budding process, which hopefully I can show you here in the next couple of slides. All right, so multiplication of animal viruses generally follows the same pattern, bacteriophage multiplication, but there are several differences. The main difference between animal virus replication and bacteriophage replication is animal cells can replicate the virus in the cytoplasm, or they can replicate in the nucleus. Of course, bacteriophages can only replicate in the cytoplasm because they do not have a nucleus. There are a few other differences like attachment of the virus. The animal virus will attach to the cell membrane because animal cells don't have a cell wall. The attachment is the same, that we have adhesions on the virus, also called ligands, which bind to a host cell receptor, and that's the molecule of the host cell the virus binds to. The penetration of the virus differs, and it happens either by endocytosis or by fusion of the virus envelope to the viral cell membrane. You should note, if you have an older copy of the textbook, that the author may have said that the virus replicates by or enters the host cell by pinocytosis. That is not correct. The virus would enter the cell by endocytosis, not pinocytosis. And of course, it can enter by fusion of the viral envelope to the cell membrane also. This is showing you penetration by endocytosis, where the virus binds to the host cell, and then the host cell begins invaginating again, and then it'll actually pinch off here, reforming the cell membrane, and then forming a organelle membrane around the virus, and then that will be inside the cell, and it has to be, the organelle membrane has to be degraded, as well as the capsid protein coat has to be removed to release the viral genome from the virus, the animal virus. Let me show you about fusion. Time I need to do it twice. I don't know why that changed on me, but there was an upgrade and that's been changed permanently now. So here we have a host cell. Make it like that. And actually, let's not make it that big, sorry. Scratch that. I need to show you. Let's just close paint down, restart it. There we go. This is the viral cell membrane, which of course has a double membrane. Let's go ahead and put that in. And then we have the virus attaching to the host cell receptor. Oops, that's not right. I want it here. And this has a envelope. So I'm going to put a double membrane of the envelope. I won't draw the core, but that'll be in here as well as the genome. What happens with fusion is that these two actually fuse together. Actually, I actually have it shown there. That's fused there. This fuse is here. Let's make this a little bigger. And then this fuse is here. And that fuse is here. And that's just by the fact that both are a lipid bilayer. And that obviously is touching and it can fuse together. Fuse with this over here. Trying to show it. All right. And you can see by the way I've drawn it that the virus, which would be here, can now enter the cytoplasm. Any question about any of that? Anyways, that's fusion of the envelope of the virus to the cell membrane. Encoding happens similar to bacteriophages. Biosynthesis happens similar to bacteriophages, with the exception that the virus of an animal cell can replicate in the nucleus, or it may replicate in the cytoplasm. The maturation of the viruses happens similar to bacteriophages. Any question about any of that? If not, I think I'm going to stop here and I'll continue here in our next lesson, which will be next Tuesday after Thanksgiving. Any questions? All right. Those of you who have questions can show up in the lab at 6:30. I'll only be in the lab from 6:30 to 6:45 or until the last question is answered. That's it. Any questions before I go?